Welcome to Module 5 of Cells, Tissues and Regulation. In Topic 5.1, we will learn about synaptic functions. During the course of this video, you can open your workbooks to Module 5 and try and answer as many questions as you can from Topic 5.1. The learning objectives for this topic are: what are the differences between fast and slow cell-to-cell communication? What is a synapse, and what are the structural and functional differences between chemical and electrical synapses? What are chemically gated ion channels? And what factors affect the size of the graded or postsynaptic potentials? What are the differences between excitatory and inhibitory synapses? We need to develop an understanding of neural integration and its control of neuronal firing, and what spatial and temporal summation are. We need to understand that there are a number of different sorts of neural circuits and what each one has in terms of function for the body. We need to describe the detailed steps at the neuromuscular junction, the neurotransmitter released, and the function of acetylcholine esterases. And finally, we need to describe the details of how skeletal muscle function is affected by Myasthenia Gravis, curare, and botulism. Single-celled organisms often communicate with other cells by having signaling molecules diffuse between them. Diffusion is an adequate form of communication for some primitive life forms because they consist of relatively few cells. However, multicellular organisms require more sophisticated communication systems to coordinate the activity and function of cells in organs and systems that are separated by long distances. Both the nervous system and the endocrine system are responsible for long-distance communication in the body. When we compare the signaling between these two communicating systems, there are some major differences in the anatomical arrangement, speed, and function. The anatomical arrangement of the nervous system is wired, whereas the endocrine system is wireless. The nervous system uses neurotransmitters as chemical messengers, while the endocrine system releases hormones into the interstitial fluid, which are then circulated by the blood. The speed of response of the nervous system and the endocrine system are different as well. The nervous system is a fast cell-to-cell communicating system, where messages are sent as rapid electrical and chemical signals. Therefore, the speed of the response is within milliseconds. In the endocrine system, hormones must be transported through the blood, and therefore the speed of the response is slow and may take minutes or even longer. The difference in the speed of response between these systems also relates to the duration of the response, meaning how long it lasts. The duration of response for the nervous system is also within milliseconds, whereas the effects induced by hormones can last a few minutes, hours, or even days. Overall, the nervous system is a fast cell-to-cell communicating system with rapid, precise, and short-acting functions. The endocrine system, on the other hand, has a longer duration, and the effects can be marked and widespread throughout the body. As mentioned previously, a synapse is a junction or a gap that mediates information transfer from one neuron to the next, or from one neuron to an effector cell in the form of an electrical or chemical signal. The neuron delivering the signal is called the presynaptic neuron, while the neuron or the cell receiving the electrical signal is called the postsynaptic neuron or the target cell. Most neurons function as both presynaptic and postsynaptic neurons, and each neuron can have around 1,000 to 10,000 axon terminals forming synapses. And let's discuss these types of synapses now. The electrical synapses consist of gap junctions, like those found among certain other body cells. Their channel proteins are called connexons that connect the cytoplasm of adjacent neurons. Electrical synapses between neurons provide a simple means of synchronizing the activity of all the interconnected neurons. These junctions allow the flow of ions, small molecules, and nerve impulses directly from one neuron to the next. The neurons in these synapses are electrically coupled, and the transmission across the synapses is very rapid. However, they cannot be easily modulated. In adults, electrical synapses can be found mostly in the regions of the brain responsible for certain stereotype movements, such as eye movement. Gap junctions themselves are also commonly found in the heart and smooth muscle, allowing a synchronized electrical activity of a large number of cells. Chemical synapses, however, are the most common type of synapses. They are specialized to allow the release and reception of chemical messengers known as neurotransmitters. When an electrical signal or action potential reaches the axon terminal of a presynaptic neuron, it releases chemical signals, called neurotransmitters, from the synaptic vesicles via exocytosis into the synaptic cleft. The synaptic cleft is a fluid-filled space between a presynaptic and postsynaptic neuronal membrane. Once the neurotransmitter is released into the synaptic cleft, a receptor region on the postsynaptic neuron's membrane, which is usually located on dendrites or the cell body, binds with the neurotransmitter and converts this chemical signal back into an electrical signal. In short, chemical synapses convert the electrical signals into chemical signals, called neurotransmitters, that travel across the synapse to the postsynaptic cells, where they are then converted back into electrical signals. Chemical synapses allow a unidirectional communication between neurons. The figure in this slide briefly explains what happens when an action potential reaches the synapse. Action potentials, as mentioned before, are charges that move along the membrane of an axon. Neurotransmission begins with the arrival of the action potential at the presynaptic axon terminal. Depolarization of the membrane by the action potential opens not only sodium channels, but also voltage-gated calcium channels. During this brief time, calcium floods down its electrochemical gradient from the extracellular fluid into the terminal of the axon. The sudden influx of calcium into the axon terminal acts as an intracellular messenger, triggering the release of neurotransmitters by exocytosis. For each nerve impulse reaching the presynaptic terminal, many vesicles release their neurotransmitter into the synaptic cleft. The intensity or the strength of a stimulus is proportional to the amount of neurotransmitter released into the synaptic cleft. The neurotransmitter then diffuses across the synaptic cleft and binds to specific receptors on the postsynaptic membrane. Once the neurotransmitter binds to the receptor proteins, the receptor changes its shape. This, in turn, opens ion channels and creates graded potentials in the postsynaptic neuron. The effects of a neurotransmitter last about a few milliseconds before they are terminated by reuptake through transport proteins into astrocytes or presynaptic neurons, or by enzymatic degradation or diffusion away from the synaptic cleft. Postsynaptic membranes contain receptor proteins for chemically gated ion channels. The neurotransmitters that are released into the synapse bind with the receptors and open the sodium and potassium channels, thereby generating graded potentials. The strength of a graded potential is dependent on the amount of neurotransmitter released and how long it remains in the area. Remember that an action potential is only produced if the graded potential reaches threshold. Chemical synapses are either excitatory or inhibitory. Depending on the receptor protein to which the neurotransmitter binds and the type of channel the receptor controls, the postsynaptic neuron may be either excited or inhibited. The excitatory postsynaptic potentials are graded potentials generated from the depolarization events that occur at the excitatory postsynaptic membrane. The neurotransmitters released by the presynaptic membrane bind to the receptors on the postsynaptic membrane and open the chemically gated sodium and potassium channels. These channels allow sodium to diffuse rapidly along its concentration gradient into the cell, causing local depolarization of the membrane. The excitatory postsynaptic potentials, or EPSPs, bring the neuron closer to the action potential threshold. Currents generated by the EPSPs decline with distance. However, if the stimuli is strong and a large enough quantity of neurotransmitter is released into the synapses, the depolarization reaches the axon hillock, and this triggers an action potential in the postsynaptic neuron. Binding of neurotransmitters at the inhibitory synapses reduces the postsynaptic neuron's ability to generate an action potential. At the inhibitory synapse, the neurotransmitters released by the presynaptic neuron increase the permeability of potassium and chloride ions. The ion channels allow potassium ions to move out of the cell and negatively charged chloride ions to move into the cell along their concentration gradient. As a result, the postsynaptic membrane gets hyperpolarized, meaning the membrane potential increases and is driven further away from the axon's threshold, meaning the neuron becomes less and less likely to fire. These changes in potential are called inhibitory postsynaptic potentials, or IPSPs. Large depolarizing currents are required to induce action potentials at an inhibitory synapse. A single EPSP cannot induce an action potential in the postsynaptic neuron. Multiple EPSPs add together or summate to influence the activity of the postsynaptic neuron. Otherwise, a neuron can never generate an action potential. When two stimuli at an excitatory synapse are separated in time, resulting in EPSPs that do not add together and therefore cannot generate action potential, this is called no summation. In temporal summation, the first excitatory impulse produces a small EPSP, and before it dissipates, a second impulse releases more neurotransmitter, which triggers more EPSPs. These EPSPs summate or add to cause the postsynaptic membrane to depolarize much more than it would following a single EPSP, resulting in the greater potential reaching the threshold and generating an action potential. In temporal summation, the frequency of impulses is rapid. In spatial summation, several EPSPs are simultaneously generated at various excitatory synapses, which all add up to produce a large EPSP that is enough to reach the threshold and generate an action potential. Inhibitory postsynaptic potentials also summate both temporally and spatially. Summation of IPSPs result in a greater membrane hyperpolarization, making it even harder for the neuron to generate an action potential, even in the presence of an EPSP. However, remember that it is the sum of all EPSPs and IPSPs that reach the axon hillock that determine whether an action potential will be generated or not. If the stimulator effects of the EPSPs dominate the membrane potential enough to reach the threshold, the neuron will fire. If summation yields only subthreshold depolarization or hyperpolarization, the neuron will fail to generate an action potential. Billions of neurons in the CNS are organized into neuronal pools. These functional groups of neurons integrate incoming information from receptors or from different neuronal pools and then forward the processed information onto other destinations. The patterns of synaptic connections in neuronal pools, called neuronal circuits, determine the pool's functional capability. Several neurons link together to form neural circuits that perform specialized tasks, including reflexes. Signaling within the circuits can give rise to higher cognitive functions such as thinking. And since the circuits are needed for even the most basic functions, it has been suggested that the functional unit of a nervous system is a group of neurons rather than an individual neuron. Neuronal circuits can be classified into four basic types. In the diverging circuit, input from one neuron is sent to multiple neurons in the same pathway. The diverging circuit is an amplifying circuit, where the signal generated from one neuron is amplified and distributed to many target neurons. For example, a single neuron in the brain can activate up to 100 or more motor neurons in the spinal cord and thousands more in the skeletal muscle fibers. A diverging circuit can be excitatory or inhibitory. For example, to walk we need to excite numerous muscle cells, and often in sequence. At the same time, though, we need to inhibit the contraction of opposing muscle groups, and so an inhibitory divergence will be required. If the impulse down from one neuron in the brain is sent to several skeletal muscle motor units in different muscle groups, it's called diverging circuits to multiple pathways. For example, when we walk, we move both our leg muscles and our arm muscles. In a converging circuit, the impulse from multiple neurons or receptors converge onto one neuron to initiate a strong response. For example, the stimuli that are generated by several receptors to initiate the sense of hunger when seeing and smelling appetizing food. In the reverberating circuit, the signal travels through a chain of neurons, each feeding back to the previous neuron. This type of circuit is involved in interpretive and rhythmic activities, such as breathing and the sleep-wake cycle. Now let's focus on nerve stimulus at the neuromuscular junction. When a nerve impulse or an action potential reaches the neuromuscular junction, acetylcholine, a neurotransmitter, is released at the junction from the presynaptic neuron. Acetylcholine binds to the chemically gated ion channels and allows sodium to diffuse into the muscle sarcolemma, causing local depolarizations called end-plate potentials. If the end-plate potential reaches the threshold, they propagate an action potential along the sarcolemma and down the T-tubules. T-tubule action potentials cause calcium to flood into the muscle and initiates protein filaments to contract. Acetylcholine esterase is an enzyme located in the synaptic cleft and on the postsynaptic membrane. Acetylcholine esterase breaks down the neurotransmitter acetylcholine into its building blocks, acetic acid and choline. This removal of acetylcholine prevents continued muscle fiber contraction in the absence of additional nervous system stimulation. Myasthenia gravis is an autoimmune disease caused by the reduction of acetylcholine receptors at the neuromuscular junction. In this condition, antibodies destroy the acetylcholine receptors. This condition is commonly seen in younger adults and is more predominant in females. Administering acetylcholine esterase inhibitors, which limit the breakdown of released acetylcholine, making it available for a longer period of time in the synaptic cleft, can treat this condition. This maximizes the effect of acetylcholine on the remaining acetylcholine receptors. Myasthenia gravis is characterized by symptoms such as drooping upper eyelids, difficulty in swallowing and talking, and generalized muscle weakness. Several toxins are known to prevent the binding of acetylcholine postsynaptically. For example, curare is a toxin of plant origin that is used by indigenous South American people to hunt. The toxin prevents acetylcholine from binding postsynaptically by occupying the receptor and thereby causing paralysis of skeletal muscles. Curare poisoning can be treated by administering acetylcholine esterase inhibitors. Botulism is a bacterial toxin that can occur in improperly canned foods. The toxin prevents the release of acetylcholine and therefore also causes muscle paralysis. The effects of this toxin include a dry mouth, difficulty in swallowing, muscle weakness, difficulty in breathing, and respiratory failure, which can lead to death. And treatment options include ventilation, surgery, or stomach pumping. From this video, we have learnt the difference between slow and fast cell-to-cell communication, electrical synapses and generation of a synaptic response, EPSPs and IPSPs, neural integration of EPSPs and IPSPs, types of circuits in neuronal pools. We've learnt about the neuromuscular junction and the neurotransmitter release and the function of acetylcholine esterases. And we've also learnt about skeletal and muscular function and how it's affected by myasthenia gravis, curare, and botulism.